Okay, I'm going to be talking about something very different. So this is about phylogeny estimation, which is essentially evolutionary trees. And the challenge here is that just about everything in this problem is an NP-hard optimization. And so to scale to very large data sets is very difficult. And you don't need to know any biology for the rest of the talk. But the basic idea is you want to get an evolutionary tree. This is an example of a phylogeny of human, chimp, gorilla, and orangutan. And it's saying that human and chimp have a more recent common ancestor. You might say, okay, this is only 4 species. NP hardness shouldn't be a hard problem here. It turns out that this is a hard problem not because of NP hardness necessarily, but because of the statistical model of evolution and why you need genome scale data and very sophisticated methods to be able to infer the phylogeny on just these 4 species. Okay, so why do we care about evolutionary trees? There's this very popular statement that nothing in biology makes sense except in the light of evolution, and another statement that says nothing in evolution makes sense except in the light of phylogeny. So you think of getting a phylogenetic tree as a tool to answer biological questions. Once I've said that, we're not going to talk about biology henceforth, except for some aspects about sequence evolution models. Okay, so phylogenomics is the marriage, basically, of phylogeny estimation with genome scale data. So you can think of this as big data, because genomes are very large, but it's actually, what's really interesting here is the computational aspects of the statistical estimation. So there is some aspect of this which is machine learning, but really only in the context of maximum likelihood. So there are existing models. We're not trying to learn models from data. We actually have models. We may be trying to learn the parameters of the existing models. So I use Blue Waters to, this is the thing we were supposed to say, design and test algorithms for core problems in phylogenomics and its applications. So the applications include things like metagenomic sequence analysis. Okay, so these are core problems, and what I'm going to describe for you now going forward is more or less algorithms and applied algorithms. So this talk is going to be about genome scale species tree estimation. I'll start off with the statistical models and what makes things hard. And then I'll talk about some advances in reconstructing species trees, but then how they're limited by data set size and heterogeneity, and then what we've done to really scale them to really large data sets. Okay, so this is an idealization of sequence evolution from a common ancestor. So I want you to think of human, chimp, gorilla, and orangutan. And each of the leaves here is a species. It's a genome from a species. So this is the genome, or a little stretch of the genome from the ancestral species. And what you're looking at is that stretch of the genome in the species as the sequences have evolved down this tree. So there's speciation events. This is a speciation event. This species had 2 subspecies. This is the region within the genomes for those 2 subspecies. So think of this as you're looking at the genomes within the species tree. So if you notice what's going on here is the sequences are changing. The sequences are changing by the letters are changing into other letters. Now A, C, T, and G are nucleotides, okay? So it's just a string of nucleotides. The nucleotides are changing, but they're not, the sequences aren't getting longer. They're not getting shorter, they're not getting longer, they're just changing. And that is the assumption that the only thing that's happening in evolution are substitutions. This is not true. But this is the mathematical model that is basically in play. And if you open any molecular evolution textbook, this is the kind of model you're going to see. So what you want to be able to do is take the sequences at the leaves of the tree and reconstruct the tree. So already you see something strange. This is an unrooted tree, right? The thing that generates the data is a rooted tree, but what you're reconstructing is an unrooted tree. The reason for this is that the mathematical models of sequence evolution are time reversible. You can't locate the root. So you're just going to get an unrooted tree back, and then where you root the tree, you have to use additional information from biology. Okay, so how do we reconstruct a tree from sequences, right? It seems like magic. No, it is magic unless you have a mathematical model, so we're going to talk about a mathematical model. This is a very simplified model I'm going to describe right here. But again, the sequences are evolving down a tree. The positions in the sequences are called sites. The different sites are assumed to evolve under exactly the same model and independently of each other. So an IID assumption, independently and identically distributed. This is not realistic, okay? But this is what you're going to find in all of the molecular evolution textbooks. IID evolution across the sites. Some sites are slower, some sites are faster, but that's all the variation you get. And so all you need to specify for the model is on each edge, if you're changing, how you're changing, right? So the probability of changing on the edge, and then if you change, how do you change between the other nucleotides? So the simplest model is called the Jukes-Kanner model, and it assumes that every edge has a substitution probability, and then if you change with equal probability to the other nucleotides. That's the entire model, okay? So you can make this more complicated, and people do. They use it, look up to like the general Markov model. General Markov model allows a much more generalized 4x4 substitution matrix, but otherwise it's still the same kind of thing. Okay, so that's a mathematical model, and now that we have a mathematical model, and you can see that the specification of the model is almost complete except for those parameters, the substitution probabilities. So in maximum likelihood, you would have sequences, and you would try to find the best Jukes-Kanner model tree. So that would be the tree topology and the substitution probabilities on the edges that maximizes the probability of the data. In the general Markov model, you would have additional parameters to estimate. So that's the phylogeny estimation problem. Given sequences, find the tree. You can quantify error, and we do that in simulations. So we do simulations, and we quantify error. And the way you quantify error is every edge in the tree is associated with a bipartition. So for example, the edge here splits S1 and S2 from 3, 4, 5. So does that. So the estimated tree and the true tree can be compared for their bipartitions, and you can say which edges are wrong. False positive edges, false negative edges. This estimated tree has one false positive, one true positive. It's not marked. So 50% of the internal edges are wrong. So the error rate is the percentage of the internal edges that are wrong. You'd like that to be 0, okay? As close to 0 as possible. Statistical consistency is saying as the sequence length goes to infinity, will your error go to 0? So this is just a statistical question. And you can only establish this with a mathematical proof. You cannot establish it with a simulation. On the other hand, you can do simulations, and you can see how quickly the error goes to 0. And it won't go to 0 monotonically, okay? Okay, so mathematical questions. Is the model tree identifiable, which is to say, is it defined by its probability distribution on the site patterns? Which estimation methods are statistically consistent? Some are, some aren't. How much data does a method need to recover the tree with high probability? It may need a polynomial number of sites. It may need an exponential number of sites. And what are the computational issues? So those are the kinds of questions that we ask. The statisticians typically are asking the first questions, and then the computer scientists are typically asking the last question. And when I talk about performance, I'm usually talking about the accuracy issues. Now for HPC people, you're typically talking about running time and stuff like that. So forgive me, when I talk about performance, I probably mean accuracy. But the answers, we know a lot about identifiability. We know which models are identifiable, which ones aren't. We also know which methods are consistent and which ones aren't. We know a little bit about how much data methods need. And then that means analytical closed forms for how much data suffices for high accuracy. And unfortunately, the best methods like maximum likelihood are computationally intensive. And when I say they're computationally intensive, I mean, A, they're trying to solve NP-hard problems, but worse, they really don't work well on very large data sets. The codes don't work well on large data sets. Bayesian estimation is even worse than maximum likelihood. Okay, so computational issues. Maximum likelihood is an NP-hard problem. Tree space is growing exponentially with the number of leaves in your tree, which is to say the number of sequences that are given as input. So if you're trying to get a species tree on 100 species, that's one thing. Double the number of species, the running time doesn't just double, right? If you try to solve things exactly, this is impossible. Heuristics can be quite good, but none of them are good enough. Bayesian methods need to converge to a stationary distribution, and they don't. Now, the key issue here is that parallelism is not enough. It helps, but it doesn't actually change things from being impossible to being okay. Because you can reduce the running time by a factor of 1,000, and you have not changed it from being intractable to tractable. Okay? So we need actually better methods, and worse, we also need methods that can run on large data sets. And when I say large data sets, there is an idea that the true species tree will have 8 million species. We are very far from being able to deal with that scale. Okay, so genome scale data gets at part of the issue, which is, remember, statistical consistency, error goes to 0. So biologists have thought that if they could just get genome scale data, you know, this delight in having big data will solve all your problems. So does big data solve all your problems? Unfortunately, the answer is no. What I'm going to show you is it creates new problems. So phylogenomics is this idea of taking genome scale data and doing phylogeny estimation. The problem is that different parts of the genome have different trees. So human, chimp, gorilla, and orangutan, different trees across the genome. Different genes have different trees. And the reason for the trees being different are biological. These are not estimation error. This is actually different trees. One of the reasons that you have different trees is this thing called incomplete lineage sorting, which is a very dominant problem. It shows up in all sorts of biological data set analyses. And it's modeled by this multi-species coalescent, which is, again, a mathematical model. This mathematical model basically says that you have a species tree, and inside the species tree you have gene trees. And the faster the speciation events, the more likely the gene trees are going to be different from the species tree. So you have this problem that gene trees can be different from the species tree. So it happened in the plant transcriptome project I worked on. It happened in the avian project I worked on, massive gene tree heterogeneity. So you have this hierarchical model that's generating data, and you'd like to infer the species tree from this hierarchical model, from the sequence data at the leaves. So how can we do this? And I'm speeding up because I'm almost out of time, but concatenation, which is the standard technique, says just concatenate all your data into one long alignment, run maximum likelihood. Statistically inconsistent, positively misleading. You can get a tree that you have 100% confidence in that's wrong. There are statistically consistent methods. The thing that works well in practice are these summary methods. The way they work is that you take the different genes and you get trees on the different genes and you combine them. Concatenation ignores the heterogeneity. The summary methods do not. So you can do this by reversing this process, but of course you have to estimate gene trees. So Astral is a technique that solves this estimation problem. It takes gene trees and combines gene trees. It's provably statistically consistent, so that if you have true gene trees, you'll get the true species trees. It's used all over the world, and its running time grows up, it goes, you know, increases with the data set size, but it's a lot faster than other methods. This is a graph comparing the running time of the previous best method, MPS, to Astral. MPS doesn't even run past 100, and Astral can keep running, but it can fail on some data sets. It fails to complete on some data sets. So Astral can fail on some data sets. Concatenation is not consistent, and it absolutely fails on large data sets. So Astral is polynomial time, but the running time grows. Okay, so this is work by Erin Malloy, who is a graduate student of mine. She's going to graduate next year. She's looking for a job, and, you know, she's great. She developed two different techniques. NJ merge is one, tree merge is another. They are ways of scaling methods to large data sets. And what they do is divide and conquer. Here's the picture. You take a data set of species, you divide into disjoint sets. You get trees on the disjoint sets using the whole genome data. You also get a distance matrix that relates all the species, and you combine them together. And so that's a way of taking a very hard problem that things don't scale in terms of the number of species, divide it into small subsets, analyze the small subsets, construct the trees, combine them. What's really cool is that it improves things. So this is a comparison of running Astral by itself or in the pipeline. The top is the error, the bottom is the running time. And what you can see is that blue and yellow are the two variants. Yellow is Astral by itself. Blue is Astral inside the pipeline. And there's no error. There's no change in the error. But the running time is dramatically different. Astral can actually fail to run on a lot of these data sets. And when it does run on, this is the 1,000 species, 1,000 gene case, the running time is here for Astral by itself, and sequential here, parallel there. So it's a dramatic difference in the running time without a change in accuracy. Concatenation, a similar story, except you can actually make the accuracy better. So reduction in the running time and improvement in accuracy. So that's what's happening with these divide and conquer strategies. They're actually improving things. Summary, the best tree estimation methods are computationally intensive. Tree space grows exponentially. Statistical consistency is a nice theoretical property, but it doesn't actually predict relative performance. Parallel implementations are helpful, but they don't actually change things from being impossibly hard to being tractable. And divide and conquer is an algorithmic design technique that can maintain statistical consistency, can be polynomial time, and can dramatically change what's possible. And divide and conquer is also highly parallelizable. Okay, so what Blue Waters enabled? It actually enabled us to design algorithms and test variants of algorithms. We can't do this without doing a lot of analyses. Each of these analyses can take months to years of CPU time for a single data set. We need to run many different variants of the algorithms on many different data sets to be able to figure out what to do. And future projects, biologic projects, will use these software. So phylogenetic inference is really interesting, and acknowledgements, my former PhD student, Siavash, and my current PhD student, Aaron, who did the work, and of course, Blue Waters. Thank you.